Hi students, this is A. Mandri Kumar Rajesh. In this video, we are going to discuss the topic called Tools for Genetic Engineering. Generally, genetic engineering is the manipulation of the genetic material to achieve the desired goal in a predetermined way. So, in order to generate more number of recombinant DNA molecule, certain types of basic tools are required for that process. So that basic tools are enzymes, vectors and host organism. So the most important enzymes which are required for the genetic engineering are restriction enzymes, then DNA ligase and alkaline phosphatase. So in this video, we are going to concentrate on restriction enzymes. So there are two types of restriction enzymes which can restrict the growth of bacteriophage, which are found in the bacteria E. coli. So in E. coli, there are two types of enzymes which can restrict the growth of bacteriophage. Bacteriophage is nothing but a virus. So bacteriophage. So it was discovered in the year 1963 by the scientist Werner Arbert and Daniel Nathans. So two enzymes are present in or found in the E. coli which can restrict the growth of bacteriophage. So if a virus infects a bacteria, the bacteria counteract the virus by producing these two enzymes. For example, this is a bacterial DNA. Please consider this is a bacterial DNA. This is a viral DNA. Then you may think how that enzyme especially make a cut on the viral DNA. How that enzyme recognize the viral DNA? Because the bacterial DNA has methyl group. So the bacterial, that is the host DNA, which contains the methyl group. So that enzyme can recognize the methyl group and it won't make cut on the DNA which contains the methyl group. So it is a viral DNA without methyl group, So that enzyme can make cut or split on the recognition side of the viral DNA. This is how the two enzymes which restrict the growth of the bacteriophage. So the bacteria which contains the methyl group in its DNA. So that enzyme can recognize the methyl group and it won't split that bacterial DNA. So it can affect only the viral DNA because it does not contain the methyl group. That is why the two enzymes are called as restriction enzymes, because it restricted the growth of the bacteriophage and it make a cut on specific site. That enzyme make a cut or split on a specific region or specific site. That is why that region is called as restriction site. Then there are two types of restriction enzyme based on their mode of action. So we can classify the restriction enzyme into two types based on their mode of action. They are exonucleases. Then the second one is endonuclease. endonuclease. So first we must understand what is called nuclease. What is called a nuclease? So nuclease is an enzyme which can break down the chemical bonds between the nucleotides, especially the phosphodiester bond. So nuclease is an enzyme which are able to break down the phosphodiester bond between two nucleotides in nucleic acid. That is called nucleases. That is called the nuclease. Then I am going to tell about what is called exonuclease and what is called endonuclease. Let me teach you the types of restriction enzymes. There are two types of restriction enzymes. They are exonuclease and endonuclease. So the exonuclease is a nuclease enzyme which cleaves chemical bonds between nucleotides at the 3' end or the 5' end of the nucleic acid chain. So this is a double-stranded DNA. So this is the 5' end and ends with 3' end. Then the 5' end pairs with the 3' end. Here the 3' end pairs with 5' end. So the double strands are connected by the bonds. The nucleotide sequence are connected by the bonds. Then if the exonuclease act on the DNA strand and it makes cut at the end of the nucleic acid. So it make a cut on the end of the nucleic acid, whether it may be a 3' end or the 5' end. So exonuclease cleaves chemical bonds between nucleotides at the 3' end or the 5' end. So it make a cut or cleavage on the end of the nucleic acid, the end of the nucleic acid. That is why they are called as exonuclease. That is why they are called as exonuclease. So it makes the cut on the outer side. On the outer side, that is why they are called as exonucleases. Then the exonucleases are important in DNA repairing. So after the DNA replication, the DNA has to proofread by some enzymes. So some enzymes proofread the DNA. So meant for proofreading. At the time, the exonuclease is an enzyme make a proofreading. So if some unwanted nucleotide sequence are present in, or a deficiency nucleotide sequence are present in the DNA, this exonuclease make a cut and it remove that deficient nucleotide sequence from the DNA. So exonucleases are important in DNA repairing. Then they are important in genetic recombination. So it can cause variation and it leads to evolution. So exonucleases are important in genetic recombination. Then they are important in prevention of the occurrence of mutation. I already told you, after the DNA replication, a proofreading process takes place at the time that exonucleases removes the unwanted nucleotide sequence. So by that thing, it can prevent the occurrence of mutation in the DNA. It can prevent the occurrence of mutation. Then genome stabilization. So it can maintain the genome in stable manner, in a stable nature, because it can prevent the occurrence of mutation. Then it can repair the DNA. It can repair the DNA. So that's all about the exonuclease. In the case of endonuclease, is a type of nucleases enzyme which cleaves nucleic acid from the middle. So endonuclease restriction enzyme make a cleavage on the middle portion of the DNA or nucleic acid. That is why they are called as endonuclease, because it may cut on the middle portion, on the inner portion of the DNA. That is why they are called as endonucleases, and it makes that cleavage at a specific region. It makes the cut at the specific region or the restriction site. So it recognizes specific nucleotides, sequences of nucleic acid, and breaks the chemical bonds between nucleotides. They are also known as restriction endonucleases because it makes a cleavage on a specific site or the restricted site. That is why they are called as restriction endonucleases. Then this restriction endonuclease enzyme is widely used in biotechnology, and they are effectively used in molecular cloning. This restriction endonuclease widely used in biotechnology and molecular cloning. Then the restriction endonucleases are otherwise called as molecular scissors. Molecular scissors because it make a cleavage on the specific region. That is why restriction endonucleases are otherwise called as molecular scissors, and they are act like a foundation of recombinant DNA technology. The molecular scissors or the restriction endonuclease act like a founder of recombinant DNA technology. Then I am going to tell about the three main classes of restriction endonucleases. So please wait a minute. Let us talk about the three main classes of restriction endonucleases. There are three types. They are type 1, type 2 and type 3. Here I have written the characteristic features of the restriction endonucleases. So the type 1, a single enzyme with three subunits for recognition, cleavage and methylation. It can cleave up to 1000 base pairs from recognition site. So it can cleave up to 1000 base pairs from the recognition site. Then the type 2, two different enzymes either to cleave or modify the recognition sequence. So two enzymes are involved. That enzymes either to cleave or modify the recognition sequence. Then the cleavage site is the same or close to recognition site. That means the type 2 restriction endonucleases recognize the specific site or the restriction site very obviously. It pinpoints that restriction site. It pinpoints the restriction site. So that is why they are called as cleavage site is the same or close to the recognition site. So the type 2 restriction endonucleases are widely used in the biotechnology because it recognizes the exact site of the restriction site. Exact site of the restriction site. It makes cut close to the recognition site. That is why they are widely used in biotechnology. Then type 3, a single enzyme with two subunits for recognition and cleavage. The cleavage site is 24 to 26 base pair from the recognition site. So a single enzyme with two subunits for recognition and cleavage. Cleavage site is 24 to 26 base pair from the recognition site. So the type 2 restriction endonucleases are more user friendly because it make exact location. It make the cut on the exact location, that is called the recognition site. Then I am going to tell about how to name the enzymes, how to name the restriction endonucleases or other enzymes. Let me teach you the nomenclature of restriction endonuclease enzyme. Restriction endonuclease enzymes named by the standard procedure with particular reference of the bacteria from which the enzymes were isolated. Here I have taken E. coli restriction endonuclease enzyme for our explanation. So the first letter E indicates the genus name Escherichia. Then the genus name is followed by the two letters CO indicates the species name coli. Then the species name is followed by the letter capital R which indicates the strain. So the enzyme E. coli was isolated from the strain R. Then the strain name is followed by the Roman numerical. It indicates the order of discovery. That means E. coli is the first restriction endonuclease to be discovered. So E. coli is the first endonuclease to be discovered. Then E. coli contains two different antibiotic resistant genes, and it contains recognition site for several restriction enzymes, and it contains recognition sites for several restriction enzymes, and this sequence is referred as the restriction site, and that restriction sites are generally palindromic in nature. That restriction sites are generally palindromic nature, which means the sequence of both DNA strands at the site, it reads same. The palindromic sequence means the sequence of both DNA strands at the site reads same. So you can read from the 5' end to 3' end. Then in the second strand you can read 5' end to the 3' end. Shows the same nucleotide sequence arrangement. So capital G, A, T, T, C, then G, A, T, T, C. So this type of sequence is called as palindromic repeats. Then the cleavage produced by the restriction endonuclease enzyme is very important in the design of gene cloning experiment. Some enzymes cleave the DNA, both DNA strands in the center, resulting in the production of blend ends or flush ends, and these cuts are called as symmetrical cuts. While in the case of some enzymes, cleaves and produces overlapping and recessed ends, and that ends are called as sticky ends or cohesive ends, and these cuts are called as asymmetrical cuts or staggered cuts. So that's all about today's video. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.